Hello and welcome to Chai and Why. With me today is Professor Sudipta Maiti, who is a biophysicist at TIFR. Sudipta works on proteins and what they do. So Sudipta, apparently proteins in the brain do some funny things when we grow old.
Yes, they indeed do, and they cause such dangerous diseases such as Parkinson's and Alzheimer's that many of us have heard of, and some of the unfortunate ones may have had a family member who is suffering from one of these. Now, these diseases are, as of now, unsolvable. We need to understand them better, but they are also teaching us something new about proteins that we haven't had any chance to know before. We typically think proteins are good guys. And they turn out in these cases that they can sort of turn from Mr. Jekyll to Mr. Hyde. So this is the story of understanding how proteins can change their shapes and then finally gang up together and attack the brain cells of the body. So we are very far from knowing exactly how to solve it, but using laser spectroscopy, using molecular biophysics, using microscopy of cells, we have just been able to figure out how they are working. We are still far from what to do after knowing this, but hopefully, maybe in a decade or two, we'll also get there. Thank you. Of course, I, for one, worry about senility. Figuratively, not quite for my own sake, but as the scientific background behind senility. You guys obviously don't need to worry about it. Now, if you could wake up on a Sunday morning and come for a scientific talk, senility is the farthest thing from you. Okay, but I'm glad that you are here, all here, and want to hear about this. So, okay, let me ask you. Let me give you a memory test. So, how many of you remember the 4 titles of the 4 talks that have been announced by Arnab? Raise your hands. Okay. Remember all the 4 titles? Maybe I'll ask you, what are the titles? Yes?
The one after that is on solar. The one after that is on nuclear physics, something not known. The last one is on fireworks and Diwali.
Excellent, excellent. So, a lot of you would have remembered at least the gist of these 4 things. Just think of some people, okay, who could have been some of the brainiest. They go past certain age and suddenly, within a matter of a couple of years, they won't remember anything like that. They could have walked in through that door, wouldn't have remembered how to go out through the door. That's memory loss that happens not to just a few unfortunate individuals, but by some estimates, 1 out of 4 people above 80, and many, somewhat less, but many people above 65, to the extent that many of us, I would say majority of us, would have known somebody who has suffered from this. Okay, that's Alzheimer's, and that's just one of the diseases or one of the conditions that afflict us when we are older. Okay, so this is about this and asking. Just stepping back and say, wait a minute, this is not caused by a virus or bacteria as far as we know, and that has been verified. This is not just happening to the people who somehow abuse their bodies and their brains, the drug addicts and all that stuff. This is happening to otherwise healthy people who have led healthy lives. What's the point? The fact that you are asking the question is new, because we have, till about 20 years ago, accepted such conditions as normal conditions that come with old age. But then there was some obvious fact missing, which was not everybody gets it. There are 100-year-olds running marathons, giving interviews. Fauja Singh, for example, you may have seen his interviews. And there are many other such examples who are leading perfectly healthy lives with very active brains. Well into their 80s. So the fact that it can happen to only few people gave rise to the question, what's going on? And the answer that has been coming in the last 2 decades astounded us meaning biophysicists, people who have made an attempt to look at proteins. Proteins are some of the molecules that are there which make up all of our body. In fact, our body is a machine, and these are the little parts of the machine. and they're wonderful molecular machines, and they do all the things that typically you and I are doing. And we thought we have some reasonable idea about how the machines come about, how do they work, what do they look like for many of the machines. And we had a theory, very nice theory, that the machines are made by the DNA, which is passed on to us by our parents, and these machines actually have a certain shape. This shape helps them to do certain things, and this is how evolution has designed everything. So even if I didn't know how every protein worked, we had this model for each protein. Something there goes wrong, and that's the subject of today's topic. The point is, the body is a machine, and to start with, it's not so startling that, as the body ages, some things or the other goes wrong, okay? You use it, and there is wear and tear. Take a new, lovely looking car, and after some heavy use, that's what the car is. And all of us, nobody is surprised by that, okay? Heavy usage, there is wear and tear, and the car goes like that, okay? And if the body is a machine, you expect the same, or do you? Muscle, heavy use, that's what you get. There's something very startling about this, okay? The body is a machine, yes, but it's not a machine that is built by a few automotive engineers over a few years of work. It's a machine built by 3 billion years of evolution, and it responds extremely differently when something is used compared to a normal machine that we are accustomed with. The question is, wear and tear, which could have been an easy explanation for growing old, doesn't quite explain it. I could put something to heavy use, and what I get back is a better machine, okay? So why does wear and tear have opposite effect in normal machines and men? Let's have a look. I said something about 3 billion years of evolution. It's an intelligent system. It's a system that has been honed to do the stuff, not only what it can do today, but to adapt to the environment, to adapt to the tasks that are at hand to become better, okay? And that's there right encoded in the genes, okay? That some things, gene is a conglomeration of all the machines that a man can make, all the little molecular machines a man can make, and we are producing some of the machines because it's our regular work. You stress those machines, other proteins get produced, or more of some proteins get produced, so that you can almost always cope with the extra requirement. And this is inbuilt in this intelligent system. You can see this is muscle fibers under a microscope, and this is what scientists call control of things that nothing has been done to, and this is a normal muscle under a microscope, muscle fiber, and this is what happens when you exercise. Okay? You see the muscle fibers got damaged. Yes, there is wear and tear, but this is immediately after heavy exercise. Then you follow it week after week of exercising. What you see is the muscle cells. This is a muscle fiber. This is a cartoon borrowed from Medscape, and there is signaling. So wear and tear gives rise to a message to the body that this is in demand, this cannot cope, you better do something about it, and the body is fully capable of responding to the signal, sending out new cells that will grow new muscle fibers or strengthen the existing muscle fibers. This is how you get this from that, okay? So this fundamentally sets apart our bodies from our normal machine, okay? So we are able to repair our things. And every day I shaved, an hour before I came here. I get nicks and cuts. Now, if every time I got a nick and cut and that stayed with me, I would not be able to look at my face. It repairs it. It knows exactly how to repair itself, okay? And it looks reasonably fresh, at least I hope, okay? So there is a program that can repair the things. Then the fundamental question comes back, then why do we grow old and senile, okay, at the end? And this growing old is not just physical. Here is one of my childhood heroes when I was 5 years old, Muhammad Ali, okay? I think knocking out Joe Frazier here. And this is his picture a few years ago. You can't say that he he didn't exercise, didn't do something, invariably he has aged. In fact, unfortunately for Muhammad Ali, he has aged both mentally, physically and mentally. The physical aging, which is a normal part of aging as far as we understand, but I'm not going to go into that. Maybe there are some surprises there too. We would be focusing on the mental aging. He has Parkinson's-like syndrome, okay, where he has difficulty moving his hands. There is shaking palsy, that is a tremor of the hands. He cannot walk in a very coordinated manner, and this is also something that afflicts many in the old age. Not all, many, okay. So this is a problem with the brain. The brain is somehow getting afflicted by something which we do not understand. Today's talk sort of as a model use Alzheimer's, and here you can see a normal brain if you take a cross section, and this is a brain taken from an Alzheimer patient, of course, after he died, and you see there are shrinkages in brain areas that are clearly visible. And people have gone in and have seen there are particular types of neurons. Neurons are the cells of the brain that have been lost in this disease, okay? And I'm still calling it a disease. Remember, I'm not calling it just a part of growing old, because what we have found, and that's what startled us, is that what is responsible for this condition are rogue proteins. Proteins that are in me, that are in you, are the same proteins that are in this individual who had this brain, or in this individual whose portrait is depicted in the US stamp, okay? And that rogue protein, some reason, has changed over its, it's a turncoat. It's changed over its color and started attacking our own body, okay? In Alzheimer's, and this is the same story for all these diseases. Parkinson's, Alzheimer's, even type 2 diabetes, Huntington's disease, Lou Gehrig's disease, the ALS disease, all of these diseases which we couldn't figure out what is happening have finally come down to. Now there are at least 2 dozen such diseases, each of which incurable, that happen with old age, that are now known to be caused by proteins turning rogue, okay? And obviously there is a huge social cost to it. When you have somebody in the family afflicted with it, there is a huge economic cost. In fact, US, you always calculate things very economically, in the economic burden wise, and there is already 100 billions per year, in a country which of course comes to the verge of not being able to pay its debt. This is quickly going to go up to $400 billion per year, the cost of care for these individuals. And but set aside all those economic calculations, it's like losing a human being while he's still alive. It's a personal tragedy. That's very sad. Okay. To do something about it, we need to understand it. Okay. So let me tell you how we understand it. I said rogue proteins. So, and here I start walking on thin ice. That is, to start with, I'll tell you, we do not have all the answers. Even some of the stuff I am telling you today are our best guesses, okay? We could be wrong, but this is where the field is, this is what we understand, and the fact that we don't quite understand it actually makes us excited as scientists that there is something here, some deep truth here that we need to understand. So look at this. So this is my cartoon of a protein. And as I said, protein does a particular function because it has a particular shape. Hemoglobin, it has a pocket. Hemoglobin is something that carries oxygen. Oxygen goes and sits in that pocket. That pocket has been perfected by billions of years of evolution to do that job. So the shape is there to hold that molecule. So it has a particular shape. In my case, this is this spring doll. And it normally, let's say it's a protein, I'm talking about a protein called amyloid beta. It's the single protein that has been held responsible for Alzheimer's disease. As I said, it's on all of us, but it is doing something else in, this is Dr. Jekyll versus Mr. Hyde transformation, is doing something else in the brains that are afflicted with Alzheimer's. So let's say it looks like that, and there is a cell. The cell has some rings, and but this guy doesn't talk to the cell. Okay, if it talks to the cell, it can affect the cell. It can be toxic. Okay, it's happy outside, and it's nice and monomer. When I call something a monomer, that means one single copy is scooting about in this thing. There are many such copies, but they are not attaching to each other. Okay? They are happy being nicely soluble. It's like you take sugar, you put it in water, and the sugar molecules then separate out, and they are separate and happy in the solution. Something happens in this condition so that they come together. Okay? This is a dimer. The monomer is a single protein. A dimer is when two of them have decided to join hands. Okay? And not only they just join hands, their shape changes. You know, so I have shown this as turned heads, and most importantly, you'll see they had these ears which are full rings now have opened up. This is my way of depicting that something is changing. Something in the protein is changing. A protein is like a ribbon, okay, which sort of a springy ribbon. So you open it up, the spring sort of pulls itself back to some particular shape, okay, and that shape changes when two of them or more of them come together. We don't yet know whether it's 2 or 5 or 15 or what. Ultimately, a lot of them come together, but maybe the toxic effect starts maybe right from when 2 of them come together. So my way of showing it is that the ears have opened up, and now they can go and get caught in the ring that anyway protrude from cells. So this is my interaction of these proteins with some cell membrane. This is the outer portion of the cell in my cartoon, and they attach themselves to the cell, and then they do something. Okay? So the two things that happen are that they get distorted, they get sticky. We had Nobel Prizes in this field for about 40 years of understanding of proteins, which always said that you give one particular sequence, that one particular type, it will fold into one particular shape. What I'm saying right now is very different. One protein which had one shape can take another shape. This is bad news. Okay? Something that can change its shape. Suppose your hemoglobin changes its shape, it will no longer carry oxygen. But it's even worse than that. It's no longer doing whatever it is supposed to do, which in many cases we don't know, but it is doing something extra which is toxic to us. So now imagine the hemoglobin, neither it carries oxygen, but it goes and attacks your cells, maybe your muscles or heart. Then that's bad news, and that's what we think is happening. There is some evidence, not very sure, but some evidence that this is actually what is going on. This is a cartoon. The cartoon comes from something, okay? So this is the ribbon I was talking about. This we feel is the shape of the thing that is there normally, and you and me. When they gang together, this becomes this shape. Don't worry about these little knobby things. These are just different ways of depicting a protein. But look at mainly the fold of the protein. The fold changes into a very nice hairpin, except that the hairpin is a very bad news. Okay? So this transformation is the transformation of proteins that is happening in Alzheimer's. This is something that we haven't seen before, and we are trying to understand. So let's just make this assumption based on some preliminary evidence that there is change of shape, which leads to things becoming sticky and coming together, which then leads to these things going and attacking neurons. Okay? Can we test it? We can. We did a very simple experiment in the lab, in a test tube. Okay? Nobody's brain, nobody is getting harmed in this. It's a simple experiment in a test tube to test whether this hypothesis, this something that we are saying is true or not. Can we make a protein? Take this protein. Yes, we can make this protein. The protein called amyloid beta, we regularly, some of my friends in the lab, regularly make in the lab by chemistry, by chemical synthesis. We make the protein. We ask, can we change its shape? Well, we don't know the shape very well, but we can make it sticky or not. Can I turn this protein into a sticky protein? Seems like we can by treating with alkaline solution, something changes, the protein becomes sticky, or just by changing the concentration. So you batter the proteins. So this is a test tube experiment. Take them in a test tube, you batter the proteins with alkaline solution. Then wait and watch what happens. First question is, do they get sticky? Okay, do they come together? Second question is, if they get sticky, would they go and attack a cell? If we can reproduce this in a test tube, at least we know we are fundamentally understanding the thing. Okay? How do we do it? Okay? So this is the experiment. So suppose I could see, so the way we are doing it, we can put a little beacon, like a flashlight on each protein. Of course, this is a molecular flashlight, okay? And like fireflies, you see them in the dark, and suppose you made one of the most sensitive instruments in the world which can see single molecules scooting about in a test tube. Okay? You'll see lots of fireflies flying about. They're little things, they fly very fast. You know that they're little, they're small. Now suppose fireflies gang together, they become bigger globs, perhaps they will move much slower. Okay? So if this happens in the test tube, you'll see that you still see the fireflies, maybe somewhat brighter, but they're moving together slower. Okay? Because you cannot see the proteins individually and measure them. And then you put some cells, or in our case, some cell mimics. These are little balls of nanometer size that you put in that are our models for cells. If the models, so these fireflies become like these and they become slower. So they are moving about fast, they are moving out medium. And if they attach to this, they're moving out very slow. Imagine there are, say, cows, okay? Sort of covered with, I don't know, honey or something that the fireflies like. I don't really know what fireflies like. And the cows moving around in the darkness, you don't see the cows, but you suddenly see the fireflies are really moving slowly because the cows obviously move much more slowly. So this is the experiment I'm trying to do in a test tube. They're moving out very fast, I do something, do they move slower? And then I put in my cell mimics, do they move much, much slower? Okay? This is all I can do, and a simple experiment. Don't worry about the little graphs and things like that. I'll show some difference that my friend Debanjan did in the lab. So you have these proteins, and this curve, how quickly it comes down from 1 to say 0, tells me how fast things are moving. The quicker it comes down, the faster it is moving. So you put the, before the battering happens, okay, the monomers, and there is a red and the black curve on top of each other actually. The red actually shows how quickly they are moving without the vesicles, the cell models, and the black, which is on top, is with the vesicles. The answer here is, if there is no difference, that means nothing is changing. Yes, we have been able to produce a protein which behaves normally. That is, I put a mimic of a cell and it doesn't bother the cell because it's not going and attaching. Okay. Then I batter this, the proteins, make them change their shape. I really don't know how to do it, but it's like mindless hammering of the protein. We hope that you can reproduce this disease in a test tube, and we find that yes, so that things which were small and scooting about, they become medium slow. They are actually attaching with each other. So comparing this curve, red curve, and this red curve would have shown it. I didn't think of it, I'm sorry. But this is slightly slower than this. But the real change happens when you put in the cell mimics. This gets much slower. This is something called a log scale, which means that this difference looks small, this difference is actually large, and this slows down to this. So that means we had made a hypothesis about how this works, and the hypothesis is that proteins gang together, attack the cell. Fast moving, medium moving, and very slow moving. Okay? And that we can see in the test tube. Does it work really with real cells? We put only cell mimics, right? So some of my friends have done this work with real cells, and you can put these same battered things, and these are cells without the proteins which have these beacons. These are cells with the proteins, and you can see this bunch of cells. There are a few cells here under the microscope. You see lots of things lighting up like Diwali candles. So those are these proteins with flashlights which have been badgered attaching to the cells. So the same thing happens to the cells. So we understand something. Okay? Now the main question is, can we do anything about it? Okay, that's what you guys would ask. Fine, you have understood something, can you do anything about it? This is unfortunately the answer that many of you already know. There is no treatment. There is some symptomatic treatment, both for Alzheimer's and Parkinson's, which doesn't address the cause of the disease. This cells changing shape, ganging up together, attacking the proteins changing shape, ganging up together, attacking the cells. Nothing we can do about it. Nothing. We can only sort of symptomatically try to take care of some of the effects that this has, and that seems to take care of the symptoms for a brief period. But this cell destruction has been going on in the background, and you slowly but surely lose enough cells that all these symptomatic reliefs actually don't work. So for a few years, you may be somewhat better, but then the progression goes on, and you ultimately lose most of the things that most of the faculties. So the real problem is, obviously this is a huge problem, and billions of dollars of research is going into it from the biggest drug companies. Where is the problem? Why can't they find a solution to this, at least that is semi-good? I mean, come on, do something, okay? The problem is even more fundamental. They can't even tell if a drug that they are testing is having any effect or not. Because this progresses slowly, it takes years. And you have to ask the guy, hey, do you remember that? Do you remember this? Give him a questionnaire. And that becomes subjective. A doctor evaluator says, yeah, he's maybe a little better than he was a year ago, but maybe I'm not sure. How do you develop a drug with such a poor measurement of the effect? We need diagnostics. Diagnostics means, you have a cholesterol level, you go draw your blood, there is a number that comes, okay? And you say, hey, you guys, you have not been exercising, you're not taking your statin or something, or you have to increase your dose. Same thing for sugar. There is a number. You can put a drug and say, okay, is this drug working very well, somewhat well, not at all, making it worse? We need to measure something. And here, the measure is extremely subjective. The only way we know clearly how to measure things is after the guy is dead, unfortunately. If he donates his brain to science, they will cut it up, try to measure these amyloid beta levels, aggregates, and all that stuff in different places. But obviously, that's not going to help him, okay? So we need to have some diagnostics, something in the molecules I can measure these shapes, measure the way that they are coming together. Maybe some of the things that people are doing, including the experiments some of us are doing that I have shown you, will someday have at least a diagnostics. If you have a diagnostic, you can put this thing in a rat and try to reproduce the disease in the rat, measure something, try 100 different medicines, and you'll get somewhere. The way you have to approach the cure is these two. Either you change the shape, because we think the shape change is the main characteristic that starts everything, or you break the association. They're coming together, put something in, okay? Put a wedge in and drives out. And when I put a wedge in, is actually one of the strategies we are thinking about right now, okay? We roughly know how they're coming together. We are thinking of a molecular wedge that will go in and actually pry it apart, okay? These are the things that you think of. Of course, you have to think of 100 such things, and you're lucky if one of them comes good. So these are the two possible approaches, and you now understand that we are very far from actually solving a disease in a human being. You are just thinking in molecular terms in a test tube to do something. We have got some hints, okay? These are like little lights in a very, very dark jungle. One light that we found was that zinc ions can bind to these proteins and do something so that they no longer attack the cell, okay? You'll jump up and say, oh, that's a cure. That's very far. Okay. What you have to see is whether zinc ions, because you cannot deliver zinc ion easily to the brain, and the zinc ions, if you deliver too much of it, there is a problem and all that stuff. But in a test tube, this is a big hope, because if something is going part of the way where you want to go, you try to understand how it is doing it. If you understand how it is doing it, you might be able to design something that is much better in doing the same thing. So here are some cells again. You can see some, these are real neuronal cells taken from a rat, not a human. And we put some amyloid beta, this is the bad protein, and they're all dead. This pink means they are dead. You put some zinc, and they're not dead, okay? This is a simple experiment again, and we are trying to see if we can answer these two things for zinc. Does it change the shape, or does it change the stickiness of the two things? What is it doing in a molecular way, okay? If we, with that understanding, we'll be able to design it molecularly. So some of my friends, Madhu and Venus Mithu, who is sitting at the back somewhere, they look at these, they have these things called NMR spectroscopy. They can look at nooks and crannies of these little molecules. They'll spend days looking at a little peak somewhere, whether it's going up, going down or something, ultimately they're trying to understand where does the zinc go and attack this protein that I have shown you. The recent things that they have come up with is that it goes and attacks in a particular place where the protein sort of comes together, two arms of the protein come together, and there is a sticky patch. Okay? It has a funny name, it's called a salt bridge. Okay? It has nothing to do with the sealing. It's like Na plus Cl minus binding to make salt, so the name comes from that. They think it's attacking there. Okay? So the point is that maybe we have some understanding where to attack the problem, and where we are right now, in August 7th, 2011, is that we are asking, what is it exactly doing by attacking there, and can we do that better? Okay. So that's all we have understood till now, and that of course leaves to my last slide, which is many unanswered questions. I haven't touched upon many things I'm expecting you to ask me in the post-chai session. So this is my last slide. So the reasoning bit is fun, and that's what we do as basic scientists in Tata Institute. We are all doing basic science research, and we're not trying to find a quick fix for this problem. Many times I do get asked that, have you found a solution to a cure for Alzheimer's yet? The answer is no. I'm not even directly looking for a cure. I'm trying to understand the problem, and that reasoning bit is fun. And what we are trying to do is make molecules, sometimes change nature's design to see if it makes it worse or makes it better, make machines that can see these things, can measure things very well, test hypothesis, say, I think this is how it works, then go ahead and test it, okay? And then think hard. And that's the way of basic science, and that's what we are trying to do. This is one of the machines. I'll leave you with a picture of that in this making machines things, we have just been able to make something that's, we believe, is the world's smallest single molecule detection machine made in this thing. And I'll leave you with a picture of people who do all the work while they're thinking hard. So these are people thinking very, very hard about proteins, not in the brains, but on the table. Okay? Thank you very much. That's all from my side.